Do you remember the function of contractile vacuole? Anyone?
Exhaust water and concentration of ion in the cell.
Okay, so they adjust the concentration, right, in the cell, and especially they adjust what the water volume, right? And do you remember the keywords for that? Osmo.
For osmoregulation.
Yeah, osmoregulation. That's the keyword for. So we have seen the structure and the function, right? So this is one example we mentioned before, that we have to learn to identify the link between the relationship between the structure and also the function, right? And so last week also you also had to look at the structure for locomotion of the protozoa, right? So which structure in protozoa help them move around? Which structure? You remember?
Flagella and cilia.
Okay. And then what if they do not have cilia or flagella? How do they move? How do they move? You remember?
Using pseudopodia.
Yeah, so they use so for the species without cilia or flagella, they use what we call pseudopodia. Pseudo means phones, podia means legs, right? Pseudopodia for moving. That's very good. So this week, these lessons, we look at how they get nutrient, right, and how they get nutrients for themselves, or how they take the food from the environment, and how they digest the food. This also occurs within the cell, right? And then also have a look at the ways they reproduce. This is the main topic for this week. So the question is, like, how do they feed themselves? Right. So we are talking about like animal-like organism. It means what? Animal-like organisms. So in terms of nutrition, what does it mean? Minchi?
Can you repeat the question, please?
The question is on the screen.
They feed themselves is mean you mean is they need food from the environment?
Yes, because when you talk about animal-like organisms, it means what? They have to take what? Food from the environment, right? Okay, so we'll have to look at that then. Is it true for all, when you also went on to look at that, is it true for all protozoa that they have to take food from the environment? Okay, so actually there are two ways for them to take. So all protozoas actually, before all, right, we have to say some protozoans can photosynthesize. But this is not the only mechanism for them to have food. Even those protozoans can photosynthesize. They still need to rely on their from the external food, from the food from external environment. Okay, so I would say that most, if not all, protozoans are actually what we call heterotrophic. And you remember that heterotrophic is also the, sorry, Sorry. Okay, so heterotrophic is that we are referring to the organisms that cannot make food for themselves. They have to absorb or take food from the environment, right? Then I think it means that then it should show you that the animals or animal-like organisms we share this characteristic that they are heterotrophic. So usually when we talk about animals, we are referring to heterotrophic organisms, right? But for protozoa, right, the single-cell organisms, animal-like organisms, right, I would say that most are heterotrophic. And some of them can actually maintain both ways of nutrition. It means that they can, some of them can photosynthesize, but still they take food from the environment. We have a look at them later to show that actually some of them which can photosynthesize also need to they also have the organelles to take food from the environment. Okay, so the types of taking food from the environment depends on where they live and how they live. Okay, it can be directly from the environment or maybe from the environment of the host. It means that within they take food from the host when they are parasitic. Right? So the question is like, how do they get food into the cell? Because you see the cell is bounded by a membrane, right? So how do they take food in? Do they have arms? Do they have mouth to feed? Okay, so apart from those, protozoan can photosynthesize, right? The one that heterotrophic can take the nutrient from the environment by different ways. The simplest way, which is equal to many type of cells, right, and go through by passive diffusion. Means like maybe some very small molecules, especially ions, right? They pass through the cell membrane just by the difference of the the difference of the concentration of these, right? So passive diffusion means like the concentration from the external environment is higher than in the cell. So some molecules go through the membrane, but then you realize that actually, when for protozoans that live in water, usually the concentration of ions and small molecules in the environment is much lower than those in the cell's cytoplasms, right? So they need active transport. You know active transport? You are studying cell biology now, right?
Yes.
You know, yeah. So what the active transport means?
Are they like transfer the food to their body by using their own membrane to wrap around the food?
Not quite. Now, if you study cell biology,
You mean by the proteins channel?
Yes, or protein pumps, right, or channels, right? So on the cell membrane, there are protein channels or so-called pumps, right? And then by using energy from ATP, they can import the food particle, no, not food particle, import the molecules through the cell membrane. So from the environment into the cytoplasms. So most of the time, actually, they will have to take active transport. But then active transport or diffusion is only applicable to small molecules, maybe like glucose, maybe like ions, right? So if they encounter big food particle, like you are eating a pancake, for example, right? So what did they do? And they will go through, they will carry out a process that we call endocytosis. And then this is what Ming said just now. So they have the membrane, they cover it, and they coat it, and they bring into the cytoplasms, right? Endo means internal, right? Cytosis means the digestion in the cell, right? So the digestion in the cell, endocytosis. How do they do so? For example, we have illustration here to show you the way they take food into the into the cell, right? It depends on the size of the food particle. Okay, we can classify into 3 types of endocytosis. One, we call it micropinocytosis. It refers to the situation when they have like very, very small food particle, okay? And they form what we call the vesicle over here, right? To coat these food particle and they bring into cytoplasms, okay? That's the first step, right? So they take food in, okay? Or if the food particle is larger, they call it macropinocytosis, right? So the food particle is larger. And if they have really, really large particle or even like other organisms, like for example, like they eat bacteria, they eat others, single cells organisms, they go through a process that we call phagocytosis. And they form something really big that they call it food vacuole. So those are small. In terms of structure, that the vesicle and food vacuole is the same, but the size are very different. Depends on the size of the food they take in. Okay. Now, then the next question would be like, so where on the membrane can they take food in, right? Where on the membrane they can form the food vacuole or the vesicle, right? So it depends on the species. Some species like this Paramecium, they have a fixed location on the cell membrane. Okay, sorry. They have a fixed location on the cell membrane, which is like grooved in. And this is the location they will form the food vacuole, right? And that position for this Paramecium, they call it the oral groove. Oral means mouth, right? So the groove like a mouth. And the buccal cavity, it means the base here, right? The space here, the space within the mouth. We call it buccal cavity. Okay? So this is where they will take food in. And we can have a look at this now. Okay. Sorry. Can you see the video?
Yeah.
Yes, we can see it.
Okay, good. So can you see it clearly? There's something moving there, right? Can anyone explain it? Do you see the movement right in the middle?
Yeah.
Okay. Can you have to explain it? What happened here? Do it again. My screen is please. Okay, do it again. What is happening over here? Do you see where I pointed? What's going on here? So the location, the area here, right, is equivalent to the oral groove and buccal cavity. Okay, at the base of the buccal cavity, there's something that keep forming, and these are the food vacuole. So, sorry. So if you see something is moving here, right? So what is it? Anyone knows?
It's
If
the
it's
buccal
oral
cavity.
groove, I think it's oral groove.
And what is going on there?
The buccal cavity is transferring the food to the oral groove, I believe.
Transferring the food. Can you see any food there?
There are some sort of like small particle floating inside the bubble.
Yeah, so you can see here, right? Where does it live? It live in water, right? So how do they create the movement of water into the buccal cavity? Can you make a prediction? You see anything moving here on the fringe of the cell membrane? Something is flashing here. You see it? Over here? Do you see where I pointed? You see the pointer?
Yes.
Okay, so what is it here? What? What is it? Do you remember that last week we already studied, right? Some group have what is it here? Looks like hairs here. What is it?
Like the hair is form like a shield protect the outside of the.
This is not the hairs, right? Something look like hairs, but that's not. Cilia, very good. So these are actually cilia, right? So the cilia are moving. Now the cilia are moving and they create the water movement, and they can make the cilia moving in a way that they can channel water into the buccal cavity. And when the water flows into that, they also carry along food particle. Okay? And you see they are very hungry, right? They keep eating. So at the base of the buccal cavity, they keep forming something look like bones here, right? Right? Something look like what do you call here? Trông như bánh trôi ấy, đúng không? Okay. Right. So these are actually the food vacuoles. They form at a fixed location at the base of the buccal cavity, and the food is carrying in by the movement of cilia. Is it clear?
Yes.
Okay. So for many organisms, protozoans, right, they have a fixed location. So you can consider that actually they have something equivalent to a mouth, you know, jaw mouth. Okay, so they do have something like a mouth, right, to take food in. That is one thing, right? And then where's their stomach? We look at it later. Now we'll look at another group of organisms. If you remember the amoeba, right? Amoeba we showed last week. Also, oops, sorry. So this is an amoeba, right? You see something is moving here? So the flow, right, of cytoplasm, and they form pseudopodia, remember? Right? But over here, they form two lobes, and they started surrounding something here, over here. You see it? Something start moving. It is an amoeba is eating another single cell organisms. And you can see the amoeba is much bigger than something that is moving there, right? Probably it's another protozoa. Can you see the video?
Yes, and I just saw the single cell disappear.
No, it's moving. It's become blurred. They cannot disappear that fast because they just stopped the video. Okay, so I show it again. You can have a look. Yep. So we see that's the way they will form the vacuole. So they produce the pseudopodia to surround the food, right? And they form food vacuole. So if we break it down into a diagram, it would be something like this, right? So they form pseudopodia, right? Oops, sorry, keeps going back. And then these pseudopodia will surround the food, right? And then from there, they will form the food vacuole. Understand? Understand?
Yes.
Yes.
So you can see here, for the amoeba, right? It doesn't have a mouth, right? Or I would say it doesn't have a fixed location to form the food vacuole. So it's more like the convenience, right? Whenever they touch a food particle, right, they will form a food vacuole. Understand? It's just like if you can take food from any part of the body, right? You don't have to take the food into the mouth, right? You can just like touch the food and the food go into the body, right? So it's like the way they do it for the amoeba, right? So the next question will be when they have the food into the cells, or when they have the food vacuole into the cells, right? How do they digest the food? Do they have some mechanisms similar to how we digest food, right? And how they can manage to have that? You can see that all these are big food particles. Then when they go into the cytoplasms, right, for sure, the protozoas cannot use this big food particle, right? It's just like you are eating, right? You have to break your cake into smaller pieces, right? And go into your stomach. The stomach will have to digest it, right? To break into smaller molecules. So over here, they also have to carry out the digestion, but it has to take place within the cells. So we call that intracellular digestion. Okay, the digestion takes place in the cell, intracellular. Okay, and also we call it phagocytosis for those like digesting big food particles. Let's start from, let me show you step by step for this part. Now, when the food particle enter the cell, right, or into this food vacuole, right, there are special organelles in the cytoplasm that we call lysosomes. Okay, lysosomes is actually quite common organelles in eukaryotic organisms, even in human cells, right? The lysosomes contain digestive enzymes. You remember, digestive enzyme has to be contained in a special organelles. Otherwise, these enzymes also digest the protozoans themselves. You know what I mean? If these enzymes release into cytoplasms, the cell will die. Right? So they have to be contained in a structure that we call lysosomes. And then the lysosome will fuse with the food vacuole. So they release digestive enzymes into the food vacuole. Just like our stomach, right? We release the digestive enzymes into the stomach to digest food, right? So this happening here, right? And then thanks to these the digestive enzymes, right? They started breaking down food molecules or food particles into the smaller piece and into smaller molecules. That we call digestion, right? So when they break this thing down, right? Food digestion takes place, okay, breaking down the smaller molecules. And then the next step would be, of course, when the food molecules become smaller molecules, right? And then we see that the concentration of small molecule is very high in the food vacuole. And automatically, this small molecule will diffuse through the membrane of food vacuole into cytoplasms. And that process we call absorption, because the concentration of molecules in the food vacuole is very high compared to the cytoplasms. Easy, right? Okay, so what's next? For sure, for any kind of digestion, right? there will be something that cannot be digested. Just like when you eat, right? There will be something that you cannot digest, and you end up going to the toilet every day, right? Right? So they don't have to go to the toilet, right? But they have a process to throw away the things that they cannot digest. That we call exocytosis, right? So in the food vacuole, anything that is not digestible, right? We call it it will become, after they absorb on the food, on the nutrition from the food vacuole, right? What remains is called a residual vacuole, that it contains all the things that cannot be digested. And now the food vacuole or residual vacuole infused back to the cell membrane, and they release this undigested stuff back to the environment. Is it clear now? Is it clear now? So basically, yes. You can see the food vacuole is basically a part of the cell membrane, right? So the food vacuole actually formed from a part of the cell membrane, and after they digest the food, right, they will fuse back to the cell membrane. And then this is essentially the way protozoan digest food. And this is the main mechanism for them to get enough nutrients, sorry. So this is clear for this part? Any question for this part? No? All right.
Currently, we don't have any question right now.
Okay, good. The next question will be, after they eat, they need to do something more important and also equally important, I would say so. Right, so the survival of any species that require two things, right? Get enough nutrient, right? And then grow and build up the body. And then the next thing they have to do is have babies. Is that right? So they need to have babies.
Yes.
Yes, that's the basic, what we call the basic is requirement for life, right? Life need to goes on by reproduction, right? So that's why they have reproduce, right? Recreate the body, right? Into babies, of course. The way of reproduction in protozoan is very, very diverse. So for this part, I will show you some types of reproduction in protozoa. And you can see that the diverse, even though they are very single cells organisms, but there are many different groups of protozoans. They have different mechanisms for taking food. They have different mechanisms for moving around. They look very different, and also the way they reproduce also very different, right? So we have like either reproduce asexually. You know what it means, right? Asexually, they don't need sex. Yep. So basically, you can say, right, they don't need sex to reproduce. Different mechanisms. But there's, interestingly, even though they're single cells, right, many of them can reproduce sexually. It means they need sex. Not the kinds of sex you are talking about, you're thinking about. Okay, so sex over here, we're referring to two types of organism within the species, right? Work together. So over here, two type cells, right? Work together to form babies. We call it sexual reproduction. We will look at that to define sexual reproduction in protozoans, right? And therefore, because of these things, right, some only reproduce asexually, some can do both. It means they can reproduce by themselves or they need a partner to reproduce. Right? For asexual reproduction, right? There's also different kinds of asexual reproduction for them. Right? Most go through what we call clonal reproduction, and it
Like for the bread or?
yes, the fungus-like organism. They also have the mechanisms of budding. You have to look at that, right? But in protozoans, they also have this ability. And they have also what we call multiple fission. What are they exactly? We have to look at these things. So binary fission is just simple mitosis, right? Equal to, right? So they go through simple mitosis, and the cells they divide into two daughter cells, right? Something like this. Simple, right? So either this way or transverse division, or they can have like longitudinal division depending on the species. So, but basically, from one nucleus, they duplicate into two nuclei, and then they divide the cells, right? Divide the daughter into two daughter cells. We just call simple mitosis, right? After simple mitosis, then they have the fission or cells division, all right? We call binary fission. This is one example of the binary fission. So once again, the Paramecium, Siloflora, right? They are dividing the cells now. So these one cells, right? This is a time-lapse movie. So this is actually in the real situation, it may take longer time, right? So slowly, they constricted the cell membrane so they can separate into two daughter cells. This is how they do, right? So they keep swimming in opposite direction so they can separate the cells. So it's not like simple like we have a knife and you cut into two parts, right? It takes time so they can retain the cell membrane so the cell won't break. So that's the eventual the final process of their division of the cells, right? Can you see the video? Yes or no? Okay, good. You see it? So this is the kind of process of the cells division, right? So other types of asexual reproduction is budding. So for the fission we have, right? For the fission we have from one cells into two equal size daughter cells. So the daughter cells are equal in size. So like they split equally, right? But for budding, it's also the fission, but then it's unequal, right? So the original cells, right, retain most of the cytoplasms. Okay, and then the other daughter cells is only given very small portion of cytoplasms. And that cell have to grow in size by themselves, right? Gain nutrient, eat a lot so they can increase the size, right? So that's we call budding. Or all right. And the other type is so-called multiple fission. And it's very different, and it's a kind of very special way of reproduction in protozoa. It only occurs in some groups of protozoa. Multiple fission means like from one parent cells or one, I don't know whether a father or mother, right? Into more than two daughter cells. So the simple binary fission is from 1 into 2, but over here they be from 1 to more than 2, usually thousands. Right? So the nuclei in the nucleus in the cells, right, keep dividing, keep multiplying, right? But the cytoplasm, the cells do not divide, right? So the cell will wait until they have multiple or thousands of nuclei in the cell before they start dividing cells, right? And that is called multiple fission. So they go through mitosis several times. So from 1 into 2 nuclei, from 2 into 4, from 4 into 8, and so on, right? Nuclei. And after they have like quite a good number of nuclei, then they divide the cells corresponding to the number of nuclei they already have in the cytoplasms. Is it clear here?
Yeah.
Okay, so this is a kind of asexual reproduction as well, but it's like out of your conventional knowledge, but they usually you see one cell into two, right? Over here, like one cell immediately into thousands of cells. We call it multiple fission. And they also have different name for that. They call it schizophrenia. And that term is only applicable to a group of protozoans that we call the subphylum Right? So they go through the multiple fission, right? First, the malaria parasite. Sorry, yeah, right. They attack the liver. So usually the malaria parasite is not this one. This one is like at the end already. At first, when, for example, the mosquito bites, right, and carry out the plasmodium parasite into the blood, these parasites usually will flow the blood and they swim very quickly, and they invade the liver first, and they stay in the liver for some time. That we call the incubation period, right? They stay in the liver and they multiply in the liver cells until they have like a few thousand of them in the cells. Then they break the liver cells out. Okay. After that asexual cycles in the liver, they release into the blood of the patient, and they start attacking the red blood cells. Okay. Over here in the red blood cells, they go through maybe 1 or 2 more cycles of multiple fission. So every time they have enough, they break the red blood cells out. So they are released back to the blood again, then they start invading more red blood cells. Now we can see now. For example, if one parasite managed to go into the liver and they multiply into 1,000, let's say so, something like 1,000, right? Of course. And then that 1,000 released into the blood, each one occupy 1 red blood cells, and from that, each one of them will multiply again into 1,000. So 1,000 by 1,000 is 1 million, right? But if they go for another round of the second round of multiple fission in the red blood cell, we have from 1 million by 1,000 into 1
billion.
1 billion. Is it scary now? Right, so that's the way they can reproduce very quickly and multiply very quickly in the patient's body. Now, what is the effect? When they started attacking usually when they are in the liver, the symptoms are not very obvious because they just stay there. But when they start attacking the red blood cell, this is what happened. When they break the red blood cells to release their perozoites, right, into the blood, only until then the immune system of human body detect some parasites, right? So the immune system respond. So the patient will get fever, very high fever, in fact, because they have like millions or even billions of parasites in the blood now. Okay, very high fever, high temperature in the body. But at the same time, the patient is losing the red blood cells. I already told you last week, right? When they are losing the red blood cells, they no longer have enough oxygen carrier, because red blood cell is carrying out oxygen to different parts of the body, right? Because of a drop of red blood cells, therefore there's also the drop of oxygen level in the blood. And that makes the patient feel very cold. So very high temperature, but still feel very cold. And that's the basic and the most obvious symptoms of malaria. And that's why in Vietnamese, we call them sốt rét. That name has a reason. Is it clear?
Yes.
Okay,
Yes.
so this group is very important. Therefore, people have to, scientists always have to pay attention and to study this group very carefully. And for more information over here, right, you may think that if there's a disease, can we think about vaccines, right? Is there vaccines for malaria? At the moment, not yet. They have tried out vaccines, but it seems that they do not work very well for malaria. Right? So I think you will feel that we are quite fortunate that we can create the vaccines for COVID-19, but for malaria, it's still a problem already for centuries. At the moment, I think if you go into the websites like website by that WHO, for example, there's a special section for malaria because it's still a challenge for many countries, especially the tropical country. where there are so many mosquitoes in tropical countries, and in many areas there are malaria parasites. There are Plasmodium parasites. And then that's the challenge, right? Because we cannot kill all the mosquitoes. We don't have the ability to kill all the mosquitoes, but there are just too many of them, and they reproduce quickly, right? So, and along the way, they also carry the Plasmodium parasites. Right? So somehow they are smart enough to overcome the immune system, especially before when they invade the liver. But eventually, after several processes, these things they will form something that they've been taken back by the mosquitoes. You will learn about it in the when you look at the life cycles of parasites. Okay, now we go on to the other types of reproduction that we call sexual reproduction. Okay, so I think now, do you want to have a break now? Before we go on with the sexual reproduction?
Yes, I think a break will be good.
Yes,
Okay,
please.
so 10 minutes break.
Okay. 10 minutes.